Hello, hello, hello. So, intro to 1B. I love all my units, but I love this one because I love the labs that we do. I love the kids that we explore. I love how it kind of like starts our conversation on biology, and I love bio. So, 1B, basic bio. Now, what I will say right from the beginning, we are going to start going into central dogma, protein formation, R groups, biochemistry, a whole bunch of different things that you probably, if you're coming from CP, you probably didn't talk about. If you're coming to me from honors, you might have talked about. If you've come to me from AP, you probably have heard this, but we're going to be definitely looking at some things that you probably didn't cover there either. So you need to ask questions. If you do not ask questions, this will be the start of really hard stuff for you and a really rough experience, okay? I don't know how much more I can say this. You need to be an active learner. No ceilings, okay? If you let this pass you, it is a high-speed train, and that train does not stop. Let me say that one more time. You need to be an active learner with this. That is the end of what that can be, okay? You need to dig in, you need to digest, you need to ask questions, you need to come to office hours. If you have those questions, you need to find those answers for yourself. Ask me, because otherwise more stuff gets added, and then you have more questions, and then your questions about questions about questions, and you're just lost. All right? So let's talk a little bit about what we are going to be doing. So we will spend a little bit of time with, I guess we'll just stay with your DIMB. So this is your DIMB for this unit. So we have some different, we're going to be going into like basic biological hierarchy. We're going to be looking at kind of like how systems are organized. We're going to be kind of, again, like I said, you're going to be trying to set yourself up for success. So all of these videos that are, let me move myself, all of these videos that are over here talking about different types of note-taking, different types of study habits, making a plan for yourself, making a plan for how you are going to succeed. That's what you're doing here. We'll talk about model organisms. So we talked about model organisms a little bit without the topic. We talked about them a little bit when we were talking about preclinical trials and how they're used and why they're used and all of that. So we're going to kind of look at those items, look at what model organisms are used, how they're used, why they're used, which ones are best, why we pick one over another, all different types like that. The next big topic that we'll be getting in is microscopy. So with microscopy, we're going to learn how, hopefully you've used a microscope before, but I know, painfully know that so many of you just like get to the microscope and it's all set up for you. Yeah, no, not going to happen here. You're going to have to learn how to get to a mic and how to set it up and how to find things in focus, and things will be moving. Okay, we will be working with living organisms and they will be moving across your field of view. So you're going to need to figure out how to keep them. You're going to have to be able to select those model organisms, select those nematodes from within. While you're looking through the microscope, you're going to have to be able to select those nematodes and move them to a different plate. And the only way that you're going to be able to see them is while you are looking at them. So that is how to work a microscope. So we have a couple different activities in here where we'll be looking at slides, learning how to do the field of view, which we'll talk about, kind of like figuring out how big the field is. And once you know how big the field is, you can figure out the size of your organism, which is so important, or your cell. We're going to be looking at how to make wet mount slides, because that is very important moving forward. That is such a life skill within a lab. And then we're going to kind of just be summing that up. we'll be looking at biomolecules. So we'll be looking at all the major types, so lipids, proteins, DNA, and carbs, sugars. So we're going to be looking at how they're formed with dehydration synthesis. We're going to be looking at major components of how and why they work. So we'll be looking at that. You'll be looking at, again, what we'll be doing with this is we're going to have a whole bunch of kits. And within these kits, we're going to be exploring how these systems work and how they function and how everything kind of gets pieced together. So we'll be using a bunch of different really, truly awesome kits that help to break down and explore kind of major topics within biology, major things that happen. And then after that, we'll be towards the end and we'll be looking again at careers and bioethics. So that is kind of a major overview of what we're going to be doing. So a little bit different of a slide deck for you this year because we are not all going to be in, but you'll see that some you guys that are home, you guys are going to be doing some activities. You guys that are in school, you're going to be doing other activities. We'll be combining those together so that everyone has the full picture. So really working on learning from each other, learning from these experiences. There's going to be looms that are going to come out for you, Schoology folders that you're going to be completing activities in as we're working through, and really going to keep building through this this really super interesting unit. And I hope you like it. And yeah, basic biology, our next unit within biotechnology. See you guys soon.